So today I'll be talking about ribulose biphosphate carboxylase oxygenase, which is also known as Ribisco for short, because it's kind of a mouthful. It's a long one, but it is a protein that keeps us all alive, really. It's found in plants, well, in chloroplasts to be specific, but it's known to be wildly ineffective. So let's get into it. So basically, proteins, they all have different structures and functions. But there are also, when you get a little deeper into that, there are different protein levels. So there's primary. Um, Primary structures are the simplest. And they go all the way up to quaternary, which are the most complex. Primary structures refers to the order of amino acids that occur in the polypeptide chain. Then you have the secondary that kind of gets a little bit more complex, but not really. It's still pretty simple. They're the folds that form between the atoms of the non-R groups within a polypeptide. Then you get a little more complicated with the tertiary structures. That refers to the three-dimensional structure of a single polypeptide. And then some have quaternary structures, and they're multiple tertiary single polypeptides when they're together and they form a protein. And that's as complex as it gets as far as protein levels go. But some proteins don't have a quaternary level because they don't have a quaternary structure because it's way more complex. It's several tertiary structures that are all linked together, so it's way complex compared to other ones. But ribulose biphosphate carboxylase is one of the more complex proteins that does have the quaternary level for sure. And then proteins have all kinds of different functions, and one of the big ones is enzymes, which is what the ribulose biphosphate carboxylase is. It's an enzyme. Basically, it's the catalyst for the Calvin cycle, and it plays a big role in plant life, basically, and anything with a chloroplast life. But there are different functions to proteins. There's not just enzymes. There's structural proteins. There's receptor proteins. There are not muscle proteins. Getting all mixed up. There's a lot of different functions of proteins, though. There's a lot. So right here we have where it kind of branches off between the carboxylase and the oxygenase, because that slash at the end isn't just because it has two different names. It's because the ribulose biphosphate can either, whatever it's grabbing is what it becomes, basically. Which one of the two it decides to be. Which it's kind of hard to catch atmospheric gases because they're gases, obviously, so it's a lot harder than most enzymes would have to catalyze anything. So this is a very slow moving enzyme. But if it gets an oxygenase, if it grabs onto that atmospheric oxygen, it becomes oxygenase, and it's really inefficient for the photosynthesis itself. It's photorespiration when that happens, and so it's really inefficient, and it ends up costing carbons in the end of the cycle. But carboxylase is where instead of grabbing an oxygen, it grabs an atmospheric carbon dioxide, which is what you'd want to happen, and it makes the process a lot more, you get a lot more out of it. A lot of more energy is gained. While the oxygenase, it costs a lot more energy than you'll ever get out of it to do it. It's super inefficient. And so because because Ribulose biphosphate was it evolved so long ago that when it evolved, the world was full of a lot more atmospheric carbon, and there wasn't a whole lot of just oxygen hanging out in the air like there is now. It was mostly carbon dioxide out there. So plants kind of evolved for that kind of system where they were just like, hey, it's here, let's grab it. But now they kind of try to live the same way and say, hey, it's here, let's grab it. And they don't latch on to the right thing a fair amount of the time. So that's why plants can be really inefficient. But one thing that I think is very important is that the research that's going into this protein is basically, it's very agricultural based for the most part, because if you can stop the occurrence of it becoming oxygenase a little bit more, and you can make the occurrence of carboxylase happen more, then it becomes hugely more efficient in that aspect. One huge issue that we do have in the world is that we have an ever-growing population, and not just humans, but humans are a huge, huge part of it. They're the main part of it, but we also have a growing number of livestock and other animals because we have to feed all these people that are here. And, one argument that comes up a lot is that you can't just sustain yourself off of plants, because we can't grow that many plants to sustain the whole world. We don't have enough room to grow crops. But with the research that's going into Rubisco, more research is going into it, and we're able to make some of the crops that we, have been growing for centuries grow a lot more efficiently by genetically modifying them into just being more effective, grabbing onto more carboxylase and not so much oxygenase. And it makes the product a GMO in the end, and it's not something that's really out there yet very much, but they are working on it with a few crops like rice, things that are widely consumed. But, GMOs aren't all bad, and this is kind of one of them where they're not really messing with it too much. They're just trying to, make it more efficient so that producers can keep up with the demand, and so that we don't have people out there starving to death. So, I mean, some good stuff can come from this, tiny little enzyme that you really wouldn't think you could get out of it, because, it's basically microscopic, and it comes from plants. It's not like it's an amazing enzyme or amazing protein in the way that we think of, like, oh, a piece of steak or a chicken nugget. It does come from plants. It's not super high protein for your diet kind of thing. It's what could potentially be feeding the world in the future. This is the secondary structure for Ribulose biphosphate carboxylase. And so it just goes to show, how it be in there. And then this is the ribbon structure. So you may look at this and, if you're really familiar with proteins, say, wow, that's a big one. But this is the quaternary structure. And as you can see, there's the different ones. The different colors and such are where each one would probably be a tertiary structure. But they're all together in this big, beautiful mass, and it is tiny in reality, but compared to other enzymes, it's quite large. And, this tiny little thing could, potentially end world hunger one day. And I feel like that's something really amazing that can't just be glossed over. But, um, and scientists have been they found that E. coli can sustain the ribulose once it's removed from the plants. It's one of their bacterial hosts that they have found, so it's allowed us to be able to sequence the DNA more and just be able to research it in general in a way that's a lot more effective so that we can potentially create much more efficient plants. Mm. And a big reason that plants are the way that they are is because they didn't evolve trying to be efficient. They evolved trying to make themselves have their leaves as big as they could so that they could shade out other little seedlings that were trying to grow. They weren't looking to get, what they weren't trying to be efficient and be the best producer. They were just trying to survive and shade everybody else out. So plants are pretty crazy, and people underestimate them a lot, but here they are, full of this big enzyme that's doing a lot of work, and right now it's sustaining most of us, because even if you don't think about it, something ate a plant today, and you'll eat it one day, or you're eating that plant now. You never know. But all in all, it's the circle of life. But it is an extraordinary little protein, well, large protein. Um, and it's also the most abundant in the world. And that's because plants were overproducing it. So that's another big thing. Plants were overproducing it because it's so ineffective. That's why it's the most abundant protein in the world. That's just another fun little fact. It really is an extraordinary protein. And I'm glad that I got to research it because it's just amazing what it can do.